Hello, this is Azita. In this video, we are going to talk about the sensory physiology. What is the generator or the receptor potential? If the nerve is a primary sensory receptor, it is the generator potential and also the receptor potential are created in only one nerve. But if it will be the secondary sensory nerves, which they receive the neurotransmitter, the generator potential is created in different parts. But as you see now in the screen, the receptor dendrites, they create receptor potentials. The receptor potential can be a depolarization or a hyperrepolarization. But if it is the depolarization, we draw it like this. But if it is a hyperrepolarization, we draw it like this. And if the depolarization achieves the threshold level and also more than the threshold level, if the sodium voltage regulated gated channels exist, then we have the action potential as it has shown in here. But in here, we see a comparison of action potential, generator potential, and also the stimuli. The stimuli can change by the intensity and also the force, the amplitude, and also it has duration. It means that the stimuli can happen, then there is a time, a duration of the time, and then another stimuli can happen. In here, we have a weak stimuli, the increased stimuli, and also the a strong stimuli, and also the generator potentials. The generator potentials can be shown by their amplitude, and in here, therefore, the weak stimuli, which the depolarization is weak. So the generator potential is small with a small amplitude, then it comes to the threshold level, but there is no sodium voltage regulated gated channel, so we don't have the action potential in here, only the generator potential. The generator potential can be EPSP or IPSP. IPSP means, in fact, it plays a role negatively. EPSP, it means excitatory, it means it plays a positive role. It means depolarization, but IPSP means repolarization. And also in here, it is more than threshold level, but we don't have still the action potential in this neuron. But only the generator potential. If in the neuron we achieve the sodium regulated gated channels, then we have the action potential. As you see, for the weak stimuli, the generator potential has died out and it didn't reach the weighted regulated gated channels, and it was not so strong, so we don't have the action potential, and the action potential is shown by its frequency and also duration. As they are near each other and close to each other, it means the action potential is stronger, and also the frequency is higher. In here there is an increase, and in here there is the strong action potential. As you see in here, there is another picture about the duration and also the time and also the strong stimuli. It means that for example, amplitude is higher or a stronger stimuli, the force is higher. As you see, if the force gets higher, the stimuli gets higher too. The receptor potential gets higher too in comparison with the weaker stimuli. And then at the end, the action potential gets stronger too. It means that if we have a change in the amplitude of the stimuli or the duration of the stimuli or the intensity of the stimuli, then we have a change in the duration, also frequency of action potential. And also in the trigger zone, the receptor potentials, which are not the action potential, but only the receptor potentials. It means that in this part, there is no sodium regulated voltage gated channels. They come together and they have a summation, and then they continue and continue till they get to a sodium regulated voltage gated channel. And after that, there is an action potential, which the action potential is traveling to the axon terminal. If it reaches the axon terminal and it is strong enough to reach the axon terminal, then we have the neurotransmitter release. The neurotransmitter will go and bind other channels in here or the other receptors, which can be the ionotropic or metabotropic channels. And then we have IPSP or EPSP in the next cell. So as you see in here, the receptors can be tonic or phasic receptors. And the tonic and phasic receptors create the tonic adaptation or the phasic adaptation. The phasic adaptation is fast, but the tonic adaptation is not fast, and it is slow. The slow tonic adaptation means as much as we have the stimulation, we are going to have the action potential. If the stimulation is strong enough and if we have sodium regulated voltage gated channels, then we have action potential, and the action potential continues, and slowly and slowly it has adaptation. But the fast adaptation means that we have a stimulation still, but the action potential is going to decrease. As you see in here, the response for adaptation is fast. It means that the stimulation continues, but action potential decreases or it eliminates. It will be deleted. But for the slow adaptation or the tonic receptors, we still have this stimulation, and the action potential tries to get slower and slower during the time and slowly. But for the phasic, it is fast. It means that the stimulation continues, but the action potential, there is no action potential. For example, for the temperature, for the odor, and also maybe for the tastes, we have these phasic receptors and also the phasic adaptation. And also about the receptor field of sensory neuron. The receptor field of a neuron, for example, for the cutaneous sensation, is the area of the skin whose stimulation results in changes in the firing rate of the neuron. It means the area which the changes of the stimulation will result in the change of the action potential in the neuron, which is going to the brain. For example, in here we have the secondary receptor field. What does it mean? It means that here this big area is a secondary receptor field, and these small areas are the primary receptor field for each primary neuron. For example, this is primary sensory neuron, this is primary sensory neuron, and this is primary sensory, and each one they have their own receptor fields. And this big sensory neuron has its own receptor field, which is this big part, the secondary receptor field. It means that it is, if we have changes, for example, in the yellow part or in the blue part or in the purple part, then we have a change in the secondary sensory neuron as its firing rate of neuron. It means that we have changes in the action potential. And if I have changes in the yellow part, then in the yellow part as a stimulation, then the yellow primary sensory neuron will change, but not the secondary sensory neuron. It means that for the secondary sensory neuron, this big area is the secondary receptor field, and for each one there is one primary receptor field for each primary neuron, and the convergence of primary sensory neuron allows a stimulation subthreshold stimuli to sum at the second sensory neuron and initiate an action potential. At 3 levels we have a control on the location of something, for example on the location of pain, on the location of touch. So there are 3 ways for recognizing the location of the sense that we are receiving. One of them is the receptor field. The other is the time difference, but time difference is only for the hearing, because for the noise, we don't have the receptor field. For example, the neurons cannot directly touch the noise, which is outside in the 3-dimensional world. So we use time differences. It means that the sound reaches, for example, the right ear sooner than the left ear. So then the brain at the cortex level will analyze everything and understand where the sound is located, because for the hearing receptors we don't have the receptor field, because they cannot touch the sense, because they cannot touch the noise, because they cannot touch the 3-dimensional world. So what happens, the brain is using the time differences and then understand where the sound is localized. About the lateral inhibition is another, another way to recognize the location. And what happens, for example, if I put a needle in here, then I feel the pain, or for example, if I put a pen in here, then I feel a touch in the B point, and a little on the A and also the C point. So what happens for me? At first, the needle, or for example, the pen, which is touching the skin, then we have a primary sensory neuron. This primary sensory neuron feels this stimuli, and also this primary, and also this primary, as the A and also C part will feel the stimuli, and they will transfer a receptor potential, and finally the neurotransmitter will be released. But the neurotransmitter of the B part is more. Then it comes to the secondary sensory neuron, and in here we have interneuron at the spinal cord level, for example. For the interneuron, we have some inhibition for the neighbors interneuron, which received and had received the neurotransmitter before, and it will inhibit them, so they cannot transfer the generator potential, the EPSP, for example, the excitatory postsynaptic potential, to the next level. So what happens in here? The neurotransmitter in here will be released, but in here no, and then the brain will understand. And in tertiary neurons are located at the brain, at the thalamus, for example, and they will go to the cortical parts, for example, the primary sensory area or the primary sensory cortex or post central gyrus. So the brain will finally understand where the needle was exactly, and this is called the lateral inhibition. It means that the interneuron will inhibit the other interneurons at the spinal cord level or at the medulla oblongata level. It depends that what kind of sensory sense it is going to carry, and what kind of, for example, spinothalamic tract it is going up, or medial laminispus tract. It depends which kind of tract is going up, but all in all, at the secondary sensory neuron, at the interneuron, we have an inhibition, and it shows that it inhibits and decreases the senses which are not necessary for the brain to understand, and just bring the exact point and shows the exact point